That's it. A very good afternoon to one and all. Am I audible, sir?
Yes, I can hear you, sir. Yes.
Yeah.
Can you hear me?
Yes, sir. Thank you. Okay. A very good afternoon to one and all. I welcome you all for today's guest lecture, Stem Cell Therapy for Neurodegenerative Diseases. So this is the need of the hour for majority of the clinicians as well as researchers. Many of the things are still working as a Pandora box, which we need to open up and do a lot of research towards it in the early diagnosis or in the management, particularly in the treatment. Particularly, these things are affecting the elderly population. So initially, in the beginning, we used to think that it used to affect people of more than 70 years and other things. Now we are observing it even at the age of 60 years. So this is one of the critical area which we need to concentrate on. Regarding this, Dr. Anandh, who is a neurophysiologist by profession, and sir is the professor and program coordinator, Translational Neuroscience, Manipal Institute of Regenerative Medicine, has done a lot of research regarding this, albeit Alzheimer's disease or Parkinson's disease and various other diseases he has worked on, and he is concentrating more on the stem cell based therapies. So with the blessings of our late founder president, Sri R.L. Jalapa sir, and the patrons of our institution, Sri G.H. Nagaraja, the president of our trust, Sri Rajendra sir, our vice president of our trust, Sri Hanumantraju sir, secretary of our trust, Dr. Chandrasekhar Shetty sir, former vice chancellor of our academy, Rajiv Gandhi University, as well as Adichunchanagiri University, and present advisor of our academy, Dr. S. Chandrasekhar Shetty sir, and in charge vice chancellor and registrar of our academy, Dr. D.V.L.N. Prasad, and with the best wishes of Dr. Prabhakar K. and Dr. Krishnappa J., medical superintendent of our institution, I wish to start the webinar with a brief introduction of Dr. Anandh. Dr. Anandh, as I mentioned, is a professor and who got introduced to me through Dr. Bharat. Dr. Bharat is a good friend of mine, and presently he is working as Professor of Biochemistry at Vaidehi Institute of Medical Science. He is also a hardcore researcher, and his thrust area is also stem cell. When I approached him that I need a doyen in the stem cell research who will contribute his hardcore personal experience and knowledge regarding the stem cell therapy, he has introduced me to Dr. Anandh, and I thank you, sir, for accepting our invitation and to be part of it, and I thank Dr. Bharat for introducing such a notable person, a hardcore researcher, to be a part of our research. So at the end of this webinar, the guest lecture, so I request Dr. Anandh to be a part of our institution as well as a part of our fluorosis research and referral lab, which is identified as the notified as the center of excellence by our academy. We are doing a lot of research on the fluorosis. So to what extent the fluorosis is affecting this neurodegenerative diseases is yet to be identified, and to be done a lot of research on these things, as the majority of the population, as I mentioned in the beginning itself, the Alzheimer's disease at an early age group itself. In the Kola district, we are observing the population at 60 years itself. So this needs to be addressed, for which I request Dr. Anandh to join hands with us, and we will contribute with the fluoride estimation, population studies and other things. And we request you, with your vast knowledge and research experience in contributing the stem cell research and various other things, to formulate the treatment modalities to help the clinicians as well as the community, sir. So with this, I request you, sir, to please be with us and to join hands with us. And sir has done his Ph.D. from NIMHANS in the year 2005, and he has got a vast working experience. And his areas of research, as I mentioned, stem cell biology, stem cell research, and various other things. And going through his CV, there is lots of points to be noted with respect to sir's CV. He has got more than 25 research publications with a very high impact indexed journals. And when I went through his articles, I could see he has got a publication with an impact factor of around 9. So this itself speaks about his research experience as well as his thrust and thirst both regarding the research with respect to the stem cell therapy. He has been awarded with many awards and honors. He is a member of notable associations, including membership in Association for Physiologists and Pharmacologists. He is an executive committee member, Indian Academy of Neurosciences. He is a Summer Fellow Award of Riken Brain Science Institute, Japan, R.N. Murthy Award, NIMHANS, Gold Medal Distinction, M.Phil Neurophysiology from NIMHANS, Bangalore. He is an executive member of Indian Academy of Neurosciences, Bangalore chapter. He is a membership in, he has got membership in Society for Neurosciences USA and various other memberships he has got. So when I went through his CV, I could see that sir has got grants of more than 4 crores. So this itself says volumes about his research experience and his impact with respect to the research that he is doing. He has guided and co-guided many number of PGs as well as PhDs, and to say the major ones includes the 7. He has guided and co-guided many MSc dissertations and which have got published in various indexed journals. He has been invited as a guest speaker for many of the talks, and he has organized many workshops, and he has done a lot of post presentation at national conference. And sir has also got post presentation in international neuroscience conferences, grant writing seminar workshops. He has attended and he has contributed with respect to these things. He has collaborated with national institutes. He has got a lot of academic responsibilities with respect to the MAHE, Ph.D. course coordinator, NAC faculty coordinator, UGC inspection faculty coordinator, Ph.D. thesis evaluator of the NIMHANS, examiner of M.Phil Neurophysiology, grant reviewer of SERV. He is a journal reviewer of many notable journals. Sir, once again, I request you to be a part of our institution as well as our fluorosis research and referral lab. We request you to be with us, sir. Thank you, and I request you to please go ahead with your presentation, sir.
Thank you for that wonderful introduction. I will share some of the core research work we are doing using the stem cells in various animal models of neurodegeneration. I'll first share my slides. Can you able to see the slide?
Yes, sir. Yes, sir. Please go ahead, sir.
So today's seminar topic is on stem cell therapy for neurodegenerative diseases. There are many neurodegenerative diseases, as sir has mentioned: Parkinson's disease, Alzheimer's disease, and then hippocampal specific neurodegenerative diseases. In our lab, we mainly focus on the neurodegenerative diseases that affects hippocampus, because hippocampus has a lot of functions in our cognitive functioning, learning and memory, locomotor activity. So most of my study, the animal models what we use is of hippocampal neurodegeneration, which also get affected in Alzheimer's disease. It also get affected in Parkinson's disease at the very late stage. Even though the Parkinson's disease starts with the degeneration is substantial nigra, now there are a lot of evidence that as the disease progress, it can affect hippocampus, and they also will have a cognitive dementia like problems. So today I will be talking about the research work. And before going into, I would like to give a very brief introduction about what is stem cells. The stem, the name itself came from plant stem. So when you cut a plant, when you cut a stem, it regenerates, it gives branches. The similar way, a stem cell can give rise to multiple types of cells, and there are different types of stem cells. It comes as pluripotent stem cells, multipotent stem cells. The pluripotent stem cells can give rise to any type of cells in the body. It can give rise to bone cells, it can give rise to liver cells, brain cells. It depends upon the culture, condition, and the cocktail of the cytokines that we use. we can push these pluripotent stem cells into any type of cells we want. So that is where the overall interest about stem cells comes from. And if you see, the basic idea about stem cells came from a tragic incident in the Hiroshima Nagasaki, where a lot of the nuclear radiation exposure killed the stem cells, and that is where there are cells which are involved in self-renewal, which are involved in replenishing the dying bone cells, the blood cells. So each cells, there is a kind of lifespan. So for example, red blood cells, we know they're 120 days. So it has to be replenished throughout our life. So where they come from? They come from the stem cells. So these stem cells, pluripotent stem cells, we can push it into derived brain cells. We can push it into derived liver cells, kidney cells. So whatever way we can, as potent as it is, as dangerous as it is. It can also give rise to some of very dangerous conditions like teratoma, cancer formations. Multipotent stem cells are very limited in differentiation. They can give rise to limited number of cells, like you can push them into chondrocytes, we can push them into osteocytes, like that. So as the cells differentiate and move on, they lose their pluripotency. So in general, if you see stem cells, they have two characteristics: self-renewal. They keep their pool intact. The volume is maintained throughout the life, and they can differentiate into the type of cells we want according to the culture conditions we provide. So this generated a lot of interest in researchers as well as clinicians, so that we can derive cells rather than waiting for a donor for transplantation. We can use these stem cells. We can produce these type of cells what we want according to the disease condition which needs cell transplantations. So in general, if you see the stem cell characteristic features, they are self-renewal. They're determined by their unlimited proliferation potential, differentiate into various cell types, pluripotent, multipotency, and unipotency. The pluripotency, as I said, it can give rise to any type of cells we want. Multipotency, there is a limitation. You can push them to certain level. Unipotency, it's almost a terminally differentiated cells. These stem cells also have something called transdifferentiation. For example, if I take a bone marrow mesenchymal stem cells and put it in a brain, depending upon the niche, they can differentiate into the cell types which are there in the niche. It can differentiate into neurons. It can differentiate into osteocytes. So they have the trans differentiation potential. They can go back and start differentiating into different type of cells. Regeneration potential. They have enormous regeneration potential. The stem cell research is going on almost for past 3 decades, and we have sufficient knowledge about how these stem cells behave. What are their positive, what are their negative sides? What are the ethical hurdles? What are the hurdles in differentiating them into particular cell types? We know that, but overall, there is one conclusive output we can say, that it has an enormous regeneration potential. So if you see the stem cells, source of stem cells, from the single cell embryo till adulthood, even at the age of 60, 70 years, we can derive stem cells from human. So there is enormous source by which we can derive stem cells. And these cells can be categorized into embryonic stem cells, fetal stem cells, adult stem cells. The embryonic stem cells, for example, you can collect it from XS-IVF embryos, fetal aborted fetus. Adult stem cells, we can get it from cord blood, Vatanjali, placenta, amniotic fluid, from the extracted tooth, from menstrual blood, from the hair follicles. So we can derive these adult stem cells from many sources from a human. It can be either autologous or you can derive it from the other patient, allogenic. So coming to the embryonic stem cells, when you culture these stem cells, generally they form a kind of embryoid body. As you can see here, these are the embryoid bodies. These embryoid bodies, when you give different types of chemicals in the culture medium, you can differentiate and push them into any type of germ cells like endoderm cells, mesoderm cells, and ectoderm cells. So it can be a real source of, bank of, we can bank upon the stem cell, pluripotent stem cells to derive. However, in a preparation for transplantation, if we have 1% of undifferentiated cells, that can be a deleterious effect for the patients. It can turn into teratoma forming cells. The teratoma is nothing but you have all germ cells in a given mass. You can see hair, we can see dental, can see bone, everything will be there in that. So if you have even a 1% or less than 1% of undifferentiated embryonic stem cells for transplantation, it can lead to brain, it can lead to tumors, it can lead to teratoma formation. This is based on a very bad clinical practice trials. We come to know that these embryonic stem cells are very dangerous. They are double-edged sword. We can use these embryonic stem cells to differentiate, make sure that the cell which are going to transplant is not having any undifferentiated embryonic stem cells. Then we can go out for transplantation. So how do we identify it? Based on the antibodies. We have advanced technology where you can segregate these cells which are differentiated, which are undifferentiated based on the antibody expression. So once we can able to sort out these cells using the flow cytometer, then we can use it for transplantation purpose. Coming to the adult stem cells, it has a lot of good advantage. First of all, you can derive these cells from adult, okay, from the patient. If you want to derive, we can derive from the patient and differentiate those cells into whatever the cell types they want, and we can use it for transplantation. For example, one known clinical practice is bone marrow transplantation or corneal transplantations. Those are all adult stem cells that are much more easy to isolate. They do not form teratoma and they have multipotency. They can home to the inflammatory area and they can get attached to that area and they can differentiate and produce the cells which are necessary for that particular disease condition. The other advantage of mesenchymal stem cell, adult mesenchymal stem cell, is they are immunomodulatory. That means when you inject these cells, there is no graft rejection. So these cells can get into your system without rejection and produce the therapeutic effects. They produce lot of proteins, growth factors, cytokines, anti-inflammatory cytokines, and they can produce a lot of vesicles, which are now coming up. I will be talking towards the end of the talk. So they are more beneficial. So some of the sources of mesenchymal stem cells are bone marrow, placenta, cord blood, umbilical cord, vat and jelly, adipose tissue, dental pulp stem cells, which we are mostly using in our lab. Why dental pulp stem cell is these pulps can be derived, dental pulp can be derived from the extracted tooth. There inside the tooth, there will be a small piece of pulp, that is the dental pulp. Why we are neuroscientists are interested in is these dental pulp stem cells are basically from the ectodermal origin, from where the neurons and glial cells are derived. So if you give a conducive environment for the differentiation into neurons and glia, these dental pulp stem cells can quickly differentiate into neurons. They can differentiate it into, for example, myelin forming oligodendrocytes, which can be used for multiple sclerosis study. So dental pulp is something we are interested in. We use bone marrow stem cells from human, we use dental pulp stem cells. These mesenchymal stem cells can also be derived from lungs, spleen, skin, breast milk, and many other sources. So what are the characteristic features of MSC? How do you, if a kind of bottle of a cell extract is given to you and you have to segregate only the adult mesenchymal stem cells which do not form teratoma, how do you identify it? There are three characteristic features. The first is when you grow these cells, they have the plastic adhering property. They adhere to the culture plate. So when you wash off, the non-adhered cells are all mostly blood cells and other cells, and the adhered ones are mesenchymal stem cells. To further characterize, to further confirm, you can do flow cytometer immunophenotyping by putting some antibodies like CD73, CD90, CD105, and they should not bind to antibody which are specific for blood hematopoietic cells. For example, CD45, CD34, CD14, CD11B. These are the negative markers. They should be positive for some markers like CD73, 90, and 105. They should be negative for the hematopoietic markers. The third characteristic feature is when you put these cells for differentiation, they should potential to differentiate into osteoblast, adipocytes, chondrocytes, which we call it as a tri-lineage potential. So these are the characteristic features. First, we have to confirm that we are using mesenchymal stem cells. So based on the mesenchymal stem cells, because earlier we used to depend only on embryonic stem cells, there are a lot of ethical hurdles. You cannot get the fetus. There is, you might have heard about during the US President Bush period, there was a ban on stem cell thing because they don't agree, their religion don't agree for aborting a fetus. So there was a lot of ethical hurdles to collect embryonic stem cells. Whereas the adult mesenchymal stem cells, you can derive from any age group. There may be little bit change in the differentiation or proliferation potential, but still there are studies that at the age of 75 also they have derived these stem cells and they have shown that still they have the stemness property. So because of this easy of availability, easy of isolation, less ethical hurdles, it does not form teratoma or cancer. There are a lot of clinical trials currently going on at various trial phases as indicated in this pie diagram. And some of the disease area that are looked into, you can see here cancer, heart disease, liver, multiple sclerosis, graft versus disease, spinal cord injury, lungs, brain diseases. So lot of study now going on using this mesenchymal stem cells. There is another stem cells that I have not included in the slide is the induced pluripotent stem cells. So what we can do is you can take these adult stem cells, you can do gene manipulation and take these adult stem cells to the embryonic stage, and then from there you can start deriving whatever the cell types you want. For example, how induced pluripotent stem cells will be useful for a disease condition is where you can derive these adult stem cells from Parkinson's disease patients. You can take their cells to an embryonic stage and see what, and then you can differentiate them into dopaminergic neurons and see how they are differing from a healthy condition. So those kind of disease modeling can also be done using these stem cells, uh, which can be very helpful for screening novel drugs. For example, Sar was mentioning about the fluoride and other things. So we can study how these fluorides are affecting the neurons, which part of neurons they are getting affected. Is there any drug, novel drug we have? You try those drugs in this tissue, in the dish, we call this disease on the disc, and then we can try this novel drugs and see whether it has any therapeutic effect. If it has any therapeutic effect, what kind of mechanism, what signal transduction pathway they are activating, how we can get a kind of much more detailed mechanism of the drug which is giving this neuroprotection, we can study using this in vitro model. So coming to the research work in our lab, as I mentioned, we are focusing on stem cell therapy for neurodegenerative diseases. I have done my postdoc in Duke University and then in University of Tennessee Health Science Center. In this place, I have learned how to derive the stem cells and how to transplant these cells into the disease animal model of neurodegenerative diseases. What are the behaviors of the stem cells, whether they form any cancer-like thing, or what kind of mechanism by which they are activating the neuroprotective pathway. So we have used this thing. I will just explain about that in the coming slides. So as I said that most of our study focus on hippocampus. A brief introduction about hippocampus, that it is a part of the brain region, subcortical structure, which is very essential for learning and memory. It is involved in storing the memory for a short period of time. It consolidates the memory and then it distributes those memories to the cortical region. So it is a kind of transient part where the memory, whatever the learned memory, goes into the hippocampus, stored for some time. And there is a hypothesis that during your REM sleep, these memories are reactivated and it gets strengthened. And the memories which are not necessary for your survival, it is overloading your brain, it will be vented out as a dream. And this memory consolidation takes place during the sleep. And during the sleep, it also store these memories in various cortical regions. So when we think about an elephant in a microsecond, these memories are again pulled back to the hippocampus and given a kind of how an elephant looks like, and it can make you to react for that particular scene, whatever the questionnaire is or whatever it is. So hippocampus is very important for short memory consolidation as well as for memory retrieval. And this hippocampus is considered to be one of the kind of very early brain development, and then later the cortical layer came to protect the hippocampus like that. So as important as it is also very susceptible to various disease condition. It is also susceptible to, in Alzheimer disease, in depression, anxiety, epilepsy, Parkinson disease, viral infection, alcohol intoxicity, diabetes. In case of heavy metal toxicity, for example, we have studied aluminum toxicity, and we have seen that how it affects the hippocampus. So we have been working on hippocampus, and we are well familiarized with the anatomy of hippocampus. We are good in analyzing the different types of neurons present in the hippocampus, how it is affected in a neurodegenerative diseases, how the stem cell transplantation can protect this different types of neurons so that the memory, the higher order brain function is preserved. So why the name hippocampus is when you dissect out the hippocampus from the rat or from the mouse and place it next to a seahorse, it looks exactly like a twin brothers. So that is why the name came as hippocampus. The hippocampus, the overall idea that there is a brain area which is involved in learning and memory, involved in memory consolidation, involved in memory retrieval, all came from this gentleman. His name is Henry Molition. So when he was 7 years old, he had an accident and he had a brain injury. And following that, he started developing epilepsy, seizure. So it became worse as he became older. At the age of 25, he was totally, non-functional. Every day he used to get many episodes of seizure. He could not able to go to job. He could not able to go for his essential needs. There in abroad, you need to have a car. You need to have the ability to drive a car, but he could not able to run his day-to-day life activities. So when the doctor, the neurologist identified that his epileptic focus, the seaside focus is starting from the hippocampus. So they decided to remove hippocampus. And these two doctors, Dr. William Schofield, who was a neurologist, and Dr. Brenda Milner, who is still alive, she's a psychologist. They studied him thoroughly. The whole idea about hippocampus came from this gentleman that following the surgery of the hippocampus, removal of the hippocampus, he was perfectly fine with in terms of the epilepsy. The seizure intensity reduced. He started showing improvement in his, the treatment. There was a drastic reduction in number of episodes of seizure per day. However, he had a problem, that is he could not able to recall any recent memories. He could able to recall his old memories, but recently happened things like meeting Dr. Brenda Milner, who was studying his psychological output after the surgery. Every day he used to pretend like as if he's seeing her for first time, like that. So he started forgetting about the past, the very recent incidents. He could not able to, store those memories. But if you recall about his childhood memories, he could be able to tell that during this year of vacation, I went to my grandmother's house. I did all these things. So from this, the whole idea of the hippocampus and its function in learning and memory consolidation, memory retrieval came to picture. So based on this, lot of studies came up in monkey models, in rat models, that you can specifically damage hippocampus and recapitulate the same behavioral phenotype what Henry Molition was showing up. So we use the similar model. This is this is Henry Molition, who passed away in December 2008 at the age of 82. So based on this input, lot of researchers started using the hippocampus as the major thing to study how the brain is functioning, because the anatomy is well defined, the neuronal structures are well defined, the connectivity between different parts of the hippocampus are well defined, where the input comes, where the output goes, how the memories are stored between the synapses, what is the code they use to store for long-term and short-term memory. So a lot of these ideas stems from this gentleman's you know, condition. So we also use that kind of model. We use this rat model where we use the stereotoxic surgery, and based on the three-dimensional coordination, we can go into any part of the brain. We can lesion that brain area and study how what are the behavioral symptoms the animal is showing. There are a lot of things that we can study from the rat model. They also express cognitive function, depression, and anxiety. So those kind of things we can study. For example, lesioning a certain part of the hypothalamus, which is involved in feeding behavior. We can tell that which part. Okay, fine. I'll shut down the camera because the bandwidth may be affected. Okay, can you hear me now? Hello?
Yes, sir, we can hear you, sir.
Okay. So just this video, I'll show it in the normal mode. So the animal has to go into the arm, they should take the reward, and they should never enter into the arm where they have finished their reward. So they should remember that. That is the hippocampal function. We call it a spatial learning. It is called that. For example, when you're going to a new place, when you're going,
me,
yes.
Can you please put it in slideshow mode, sir?
Can you see now?
Yeah. Yes, sir.
Okay. So the task is that we train the animal in such a way that they go into the arm, they eat the pellet, and they should never enter into the arm which they have already finished the reward thing. So they have to remember that. So how they remember is they consider everything, the extra maze cues, which we call it, that for example, the computer here, a chair here, we keep another extra maze cues. So when you go to a place, subconsciously, we remember some of the landmarks so that next time when we come, we correlate and we can go to that exact place. So there the hippocampus is very much involved in. For example, there is a study where they have shown that the taxi driver, the hippocampus volume is bigger than the normal people. So hippocampus is involved in encoding all this information, which we call it as cognitive map. So this cognitive map encode some of the landmarks so that it will guide you through, it will take you through that particular trajectory to reach your destiny. So this kind of method we can use to study how hippocampus is affected. I'm talking only about the animal model, so that once you know the animal model, then we can try different therapeutic things in this animal model. So I'm explaining about the animal model where hippocampus can be damaged and it can show the memory deficits. For example, here you can see the black dot is a normal animal, whereas the red dot is the hippocampus damaged one. As you can see, as the days goes, The normal one can reach 90% of correct choice, whereas the hippocampus damaged one could be able to reach only 50%. Okay, so the hippocampus is very much involved in your day-to-day activity. So when the animal is exposed to that particular apparatus, it has to remember, it has to consolidate that memory during the sleep. And next day when it is exposed, it should know that this arm, which I have already visited, once I go again, I will not get any reward. So I should not go there. That is the memory. So that is the learning and memory process. So if you leave this animal, not exposed to that particular apparatus for 30 days and bring back to the apparatus, you leave the normal control rats there, they do 100% correct. Whereas the hippocampus lesioned one here I have mentioned as VSL, which is a part of hippocampus, they will not show, they only show 60% correct choice. So this is the model, hippocampus degeneration model. So we can develop this model by injecting a neurotoxin specifically to the hippocampus and kill only the hippocampal neurons. There are different types of different area subfields in the hippocampus. We call it as CA1 hippocampus, CA2, CA3, CA4, and dentate gyrus. The dentate gyrus is one specific region which has attracted a lot of attention. Is in the adult brain, there are only two brain regions that can develop neuron that can produce new neurons throughout our life. Whereas liver, we know that it has a high regenerative capacity. Whereas in the brain, in the spinal cord, the regenerative capacity is very, very less because we do not have that much pool of stem cells in the brain. So one such specific unique area is the dentate gyrus, which is also part of the hippocampus. This neurogenesis we can trigger, we can make them to produce more by giving novel drugs. And these neurogenesis are directly linked to your cognitive function. It is also linked to your mood. For example, depression, most of the antidepressant drugs, it works to enhance neurogenesis in the hippocampus. So if your stem cell is able to reactivate the stem cells, endogenous stem cells in the host, then it is great thing. Or the stem cells we are transplanting is differentiating into these newly formed neurons, then it can amplify or it can help you in cognitive function as well as in mood stability. So this model, I will talk about another model which was we go. So using this model, the very first study, in fact, in India, probably this could be the first study, which was a DBD funded project. When I was doing my PhD, I was part of this project where we transplanted cells into this animal model. So we cultured the cells, and that time there was no stem cell news, not much about it, at least in India, but we had a cell line, which is a hippocampal cell line. And this cell line was derived from mouse and it was immortalized and it can be passaged for multiple times to produce hippocampal neurons. And these cells can be transplanted into animals to study how these cells are getting integrated into the neural network. Are they getting rejected by the immune cells? Whether they are helping the endogenous stem cells to proliferate, all those things can be done. This was conducted in 2002. So this was a kind of the initial trigger for me to focus on my entire career on stem cells transplantation in neurodegenerative diseases. So in this animal model, what we did is we cultured the cells, we created lesion, animal lesion, and then what we did is we transplanted the cells in the lesion area. These animals to survive for 2 months, and then we did long behavior experiment. We sacrificed animal to see transplanted surviving. Then also we saw what effect on the endogenous neural cells, whether the neural cells they proliferate, they make more dendritic arborization. What is happening to the endogenous stem cells we were looking into. So this after that patient, you can see here, this is the normal control. This is the hippocampus lesioned one. This is the hippocampus lesioned and the hippocampus transplanted one. So here what we use is we use a method called Moore's water maze, which is a international system where we can use for studying the hippocampal function in addition to the one I've explained here the previous way. So in this system, there is a video that I have to go to a normal mode to play it. Yes. Yes. So as you have seen in this video, the animal will be left in one corner and they have to swim in this pool, and there is a hidden platform. The water what we use is a opaque water. We mix milk with this one so that they cannot see the platform. So what the animal has to do is they have to keep these clues. What you see, an arrow, a round black dot, star. They have to keep these extra maze cues, and they have to coordinate and find where the hidden platform is. So when you train this animal for 10-15 days, the first day they will make a start, they will roam around, they will find the platform. As they learn, once you leave, they quickly straight go to the platform and escape. So the time to reach the platform, latency is considered as their learning index. So here in the graph, this whole thing can be video recorded and it is a software enabled. So you can get the whole trajectory of how this animal swimmed through this Morris water maze to reach the platform. So for example, here you can see on the day one, they spend a lot of time roaming around and then they have found the platform. But as the trail gone, you leave the animal here, straight away they go to the platform in the normal control. Whereas in the hippocampus damaged one, they take a lot of time to find the platform. But in case of the cell transplantation one, like with the normal control, they could quickly go to the platform, hidden platform, and they can climb on that and show their learning ability. So this is a proof that the cell transplantation can either mitigate the neurodegenerative process. Most of the neurodegenerative diseases are progressive in nature. It is not an abrupt cell loss, just a sudden shock, something like an alcohol toxicity or a kind of heavy metal toxicity. It is not like that. It's a very slow process. It takes for years for the neurons to degenerate. So here in this case, we have triggered a degeneration and then we have transplanted the cells and we have slowed down the progression of the neurodegeneration. You place this animal in their home cage, bring it back to this Morris water maze settings. You put still the transplanted cells demonstrated the learning ability, the remembering memory ability as equivalent to the normal control animal. So this is the indication that the cell transplantation can be beneficial for a neurodegenerative diseases, and most of the neurodegenerative diseases do not have any drug available to cure them. It can only provide symptomatic reliefs, but it will not slow down the disease. It will not completely cure the disease. So stem cells probably or cell transplantation can probably slow down the disease so that they can live a healthy life. So after the animal was sacrificed, we looked into where these transplanted cells are gone. As you can see, the bright light, the small dots, these are all transplanted cells, which are labeled with a fluorescent dye, which we can visualize under the fluorescent microscope. As you can see, this is the hippocampus. This is the CA1 region. This is the dentate gyrus I talked about the neurogenesis, and the cells we transplanted, they have gone and settled in the fiber tract called corpus callosum. For some reason, these transplanted cells, they first go and settle in the fiber tracts. Probably they are getting the niche, they are getting the nutrition there in the fiber tract so that they can first survive and then migrate. So this is the first report we showed that the cells can survive, they can get integrated into the neural network, and they can migrate to various brain regions. As you can see here, the small dots in the dentate gyrus, in the other regions, these cells are migrating. You can see these lines, the cells are migrating. You can see, for example, here, From the corpus callosum, they are migrating. The zoomed one, you can see these cells. These cells, what you are seeing, they are migrating and they are coming and settling in dentate gyrus, which is the neurogenic area where the new neurons are formed. Why they are going to the neurogenic area? Because that is the environment where they can survive. They can differentiate into neurons because all the cocktails of chemicals and transcription factors are available in that neurogenic niche, so that they are preferring to migrate to the neurogenic region. As you can see here, this is the dentate gyrus where these transplanted cells have got integrated into the dentate gyrus. So this was the first evidence we showed that the cells, even though they are cell lines, they can get integrated into the nervous system. We also looked into what is its effect on the endogenous cells. For example, the patient's brain cells. Is there any effect of transplanting the external cells? Whether the external cells can influence the health of the patient's neurons. So for that, we looked into something called Golgi staining, where you can see here, this is a normal control where you have the cell body, the dendrites, the branches, axon branches, whereas in the hippocampal lesioned one, you can see there are not many branches like in the normal control. Whereas in the transplanted one, it is much more than the normal control. You can see lot of dendritic arborization we could able to see. That means the cells you are transplanting is actually making the sick neurons to become healthier. How they are producing this effect, that I'll be talking in the next few slides. So they are enhancing the survivability of the host neurons. So it is not only that these transplanted cells are differentiating into neurons or glia, but they are also boosting up the endogenous neurons to survive for longer time. That is where we are delaying the progressive neurodegeneration. So fine, we have transplanted it. We sacrifice the animal after, for example, after 6 months, and we looked for these red cells which we labeled. Surprisingly, we could not able to see not many cells as what we saw 2 months of the post-transplantation. That means these cells are not surviving for longer time. That is one side. So next is, in this study, we transplanted the cells into the hippocampus directly, but clinically it is not translationable. We can't do transplantation in every patient who comes, open up the brain and then put the cells. So we have to come up with a method where we have to do something which is feasible, which is less invasive. So the next study what we used is we try to inject cells. By this time when we came to the next project, the stem cell is well established and we are well established to culture derived cells from human samples and culture expand it, characterize it, and we can transplant. So this, between this study and the next study, it is almost 10 years passed. So the next study what we used is, anyway, hippocampus, we have seen there is a protection. Let us go into hippocampus related some neurodegenerative or neurological conditions. So in this case, what we did is we used an animal model of temporal lobe epilepsy. So we can develop temporal lobe epilepsy in this model animals by injecting a neurotoxin called kynic acid, which is an analog of glutamate neurotransmitter. We all know that glutamate neurotransmitter is an excitatory neurotransmitter. So when you give it an overdose, what will happen is it can overactivate the brain system and produce an epileptic condition. So you give an injection, the animal get into a status epilepticus condition. They show 4 stages of status epilepticus. After 45 minutes of recurrent seizure, they go into a silent phase. The animal recover, they go into a latent phase. And then after 2 months or 3 months, without any neurotoxin injection, they will show epilepsy, which we called it as spontaneous recurrent motor seizure. And that is equivalent to your patient's epileptic condition. So this goes on till the death of the animal. Some animal respond to drugs, some animal will not respond to drug. One of the difficult thing in treating temporal lobe epilepsy is nearly 30% of the patients do not respond to the current anti-epileptic drugs. They are refractory in nature. So in such patients, removing the hippocampus is one option. The another option is that we are exploring is, can stem cell can cure these people? So using this wonderful animal model, we are studying that. In this animal model, once you give this injection and the animal starts showing status epilepticus, we have sacrificed the animal at different time point to see how the brain structure is changing, what is happening
Excuse me,
inside,
sir.
because it, yes.
Sir, slides are not visible. Please share it again, sir. Yes, sir. Yes, sir. Now it is visible. Thank you, sir.
Can you see now?
Yes, sir. Yes, sir. We can see.
Okay. So we can use this animal to study the changes after the first initial injury. That is the first after canic acid injection, we get the status epilepticus. After the status epilepticus, there is a latent period where something is happening in the brain that is making the brain to produce epilepsy by itself without any external stimulation. So this latent period is very important. So if we can study the anatomical or some biochemical molecular changes happening during this period, then we can intervene at this level to prevent the development of chronic seizure. So using this model, we have sacrificed the animal. As you can see here, this is in the normal control animal. This is the normal control animal, and this is from the status epilepticus. This was sacrificed after 24 hours after the status epilepticus. As you can see, the green fluorescent, those are immune cells. We call it as reactive astrocytes. There is an enormous inflammation after the status epilepticus. The inflammation is the actual trigger for the neurodegeneration. So if you could be able to prevent this, probably we may able to prevent the neurodegeneration of various part of the hippocampus and other regions. And we can also prevent the number of the animal or the subject getting into the chronic epileptic conditions. We also confirmed this inflammation using another microglial markers, where you can see this is a normal control and this is the dark staining, whatever you see, those are the inflammatory cells. So if you sacrifice the animal and then look for any early sign of cell death, yes, there are early signs of cell death. You can see here's the fluorescent, green fluorescent one. This is a kind of staining called fluorojade staining, which will only label the cells which are in the process of death, not the dead cells. You can see here, you can see lot of cells in the epileptic animals. And then after 1 week, we sacrificed another set of animal to look for the anatomical change in the hippocampus. And you can see here, the CA3 neurons are lost, the CA1 neurons are lost, the dentate hilus neurons are lost. In the chronic condition, after 30 days, you can see even further damage. So this we call it as hippocampal sclerosis. The hippocampus becomes smaller, they become harder, and they lose lot of neurons. And when hippocampus lose its neurons, become harder, they will definitely show cognitive impairment. They will definitely show anxiety and depression kind of comorbidities. So this model is, I can go detail into this one, but for this study, this talk, I have narrowed down only to the neurodegenerative part. So using this model, we want to study whether we can inject the stem cells not into the hippocampus, not into the brain, but can we inject through intravenous mode? So it can be applicable in clinical settings. So this is again a DBT funded project where we looked into the potential of intravenous administration of the stem cells, particularly the adult mesenchymal stem cell, bone marrow mesenchymal stem cells, and dental pulp stem cells. I mentioned that why we are interested in is because it can differentiate into neurons and glial cells. So one advantage of this model is, animal model is, whenever there is an episode of seizure, we have found out that there is a leak in the blood-brain barrier, which is good because when we are giving the stem cells through systemic injection, either intraperitoneally or intravenously, there is a problem that these cells first goes and accumulate in the lungs and they get entrapped in the microvesicles. And then through the microvesicles, they pierce through and they come out and then they move to the brain region. By the time significant amount of cells are entrapped in the lungs, they can also cause embolic like effect, which is not a kind of conducive environment. So if you have a, and another thing is, even though the cells which are piercing through and coming through these microvesicles, they are less in number. Again, you have another gateway, that is the blood-brain barrier. So passing through the blood-brain barrier is very difficult. But in this case, even though the disease condition, it is a bad news that the blood-brain barrier is damaging. For a stem cell therapy, it may be a wonderful opportunity to put more cells into the brain. So we used that narrow window. We studied that after the epilepsy, after the chinic acid injection, till how long this blood-brain barrier is open. How we could be able to detect? We could be able to detect based on some of the biomarkers in the blood system, which are normally not present, almost nil. But only when there is a blood-brain barrier damage, we can detect this protein in the blood samples. So that is how we have done it, that we use these markers, S100 beta and GFAP, both are astrocyte markers, which are normally not present in the blood circulation. But when there is a blood-brain barrier damage, they comes to the peripheral circulation, which can be captured using a ELISA method, or which can be captured using a Western blotting method. So that is how we have done it. We detected that within 3 hours, we could able to detect the leakage of this protein. That means the blood-brain barrier is leaking 3 hours. That is the earlier time point we have checked. The latest time point we have checked is the 5th day. It may be beyond that also, but we want to just initiate the therapy. So we used this window. We injected the stem cells on day 3 because the animal will be very weak. We cannot handle them. So we gave them some time for recovery. We gave the injection on the day 3, and then we allowed the animal to live in their respective cage. And then we have done a lot of behavior experiments. And these experiments can detect their anxiety, their cognitive function, their spatial learning memory, their grooming ability. For example, in animal, for in human, if they are depressed, they don't take care of themselves properly. They don't groom them properly. The same apply to this animal. In a day, a rodent can spend nearly 50 to 60% of their time grooming. So they keep themselves clean, and the grooming also involved in the physiological stimulation to maintain their body thermostats. So all these things are a good indication that the animals, this may be a normal behavior that for other people, for a neuroscientist, for a behavioral scientist, it gives a lot of information about your brain functioning. So we can use this grooming behavior as an indication that the animal is coming out of depression, or is whether it is going through a depression phase. So we have done a lot of things, and we have gone through all this thing. Recently, we have published this paper in one of the reputed journal. So the sacrificing this animal, we came to know that, as you can see here, the chinic acid treated group, you can see these glial cells, which are exactly like, stars kind of thing with the more branches. Whereas here you can see those similar cells, but their structure is different. Here you can see, for example, this cell, this is a typical inflammatory cells. Whereas here, these glial cells, it is in a normal condition. The glial cells are very important for your central nervous system function. Initially, it was thought that the glial cells are of no use. They are just a supportive cells. The glia itself meaning is glue. It's just a supportive cells. It makes the neuron to stay in its place, so that it won't move. That was the initial belief. But now we know that the glia is, in fact, the mother of neural stem cells. The neural stem cells come from glial cells actually. The glial cells are the one which gives rise to different types of neurons. So the glial cell have multiple functions. It involves in neurotransmitter reuptake. It involves in clearing the potassium ion when the action potential is happening, so that the neighboring neurons are not getting affected, which is called as potassium siphoning, which takes the potassium and throw it in a different area where there is no neural network activities necessary. There are many functions. It provides growth factor for your stem cells to survive and differentiate. At the same time, these glial cells also do immunosurveillance. They are the immune cells in addition to microglial cells, which can rampify, make more processes, and show that as a reactive astrocytes. So when we transplant this dental pulp stem cells or bone marrow mesenchymal stem cells, we saw a drastic reduction in the inflammatory status. We also process these sections for looking into the hippocampus structure. As you can see here, These are sham control. Their sham control means they have gone through same process, but no toxic treatment. Whereas the kynic acid group, as you can see, the part of hippocampus, the complete CA3 neurons are gone. They are degenerated immediately after your status epilepticus. So we know that now after an initial precipitating injury, either through the status epilepticus or through brain inflammation, brain infection, cancer, or high fever, you get status epilepticus. For example, kids, they get febrile seizure. There is a possibility that they may undergo neurodegeneration if they are not treated well ahead. So these laws cannot be replaced. You cannot bring back these neurons. You can transplant cells so that they can cover up this area. But our strategy is not that. We want to prevent this neuronal loss by providing cells from outside. The cells can go and integrate into the network. The cells can produce very essential therapeutic proteins so that the neurons are not dying. That's why we call our study as neuroprotection. We protect the neurons from degenerating. So in both the cases, DPSC as well as BMMSC, we found there is a neuroprotection. In other brain region also, we confirmed that there is a neuroprotection. And this is an amygdala. This is a part of brain region which is involved in emotional learning, emotional aspects. It is involved in anxiety, and we have seen that there is a neuroprotection. We did a lot of behavior experiments. We found very positive results. And some of these are like, for example, cognitive tests. This is the kynic acid group, which demonstrated impairment in cognition, whereas after transplantation, we have seen the effect. There are mild difference between DPSC and BMMC, which I am not going into detail here. For example, here I have told about the grooming, how important indicator it is to tell about the brain health. So in kynic acid, in epileptic animal, there is a drastic reduction in the time spent in grooming or the count of grooming, whereas after transplantation, it is maintained. Okay, there is a difference between DPSC and the BMMSC in their therapeutic effect. And then, for example, you can detect the depression by using a method called force film test, which is commonly used in pharmacology area, where we can detect the depression-like thing. Most of the antidepressant drug screening, they use this kind of model. We have used that to show that the depression kind of comorbidity is normalized in the transplantation group. Okay, fine. Again, in this case, we have published now, it is available online. We have mentioned in the paper that after 90 days of transplantation, we have hardly seen 5 to less than 5% of these cells surviving. They survive only in the neurogenic region or in the fiber tract. That too, they survive only in a degenerative condition. In the sham control, they do not survive. That means that for the transplantation cells to survive for some time, there should be an inflammatory milieu so that the cells can be attracted to that region and it can sense that milieu and produce the proteins and the cytokines and many biological factors depending upon the inflammatory milieu. In the sham control, we could not able to see, but in the degenerative condition, yes, we have seen, but extremely limited number of cells we have seen. For example, when we have transplanted 1 lakh cells, we have seen less than 5,000 cells. But in spite of all these things, in the both the study as well as from many studies throughout the world, they have shown that there is definitely a cellular, molecular, and functional recovery after cell therapy. So this trigger our next project, that what is that it is protecting, it is inducing this functional recovery. It is inducing this anatomical recovery, biochemical recovery, molecular recovery. So there are multiple hypotheses how the stem cells can work. The earlier belief, for example, during my PhD, what I was doing at the time, the strong belief was that these cells may be differentiating into neuronal cells. The neuronal cells may be neurons, glial cells, oligodendrocytes, microglial cells. And then the another hypothesis, after some years, after half a decade, came something called secretome hypothesis, that the stem cell not only transdifferentiate, they also sit there like a drug factory. They study the disease condition and produce proteins, therapeutic proteins that neutralize the toxic environment so that your endogenous cells can survive, they can regenerate. The third hypothesis is that these cells, stem cells, which are basically a baby cells. So baby blood have much more energy than an adult. They have much more energetic thing. So these baby cells, once you transplant into the brain, they can donate some of the crucial organelles like mitochondria and other substances to the dying neurons so that the dying neurons can survive. The cell-cell fusion theory is still under investigation, whereas the secretome thing, it has really gone to a clinical level. And a lot of companies are now coming up using the stem cells as a drugstore. You trigger in vitro and you can produce the essential protein that you can collect the subject. So that is what we have done. The cell transplantation, the differentiation hypothesis, almost coming to a death because many studies, thousands and thousands of studies now coming up telling that the transplanted cells do not survive for more than 1 year, even in a human subjects. So it is the secretome based thing. So based on that, we want to study. So we initiated this study long back. What we did is in an animal model of hippocampal neurodegeneration, we created a two group where in one group we transplanted the DPSC or BMMC directly into the hippocampus. Whereas in the other group, what we did is during we culture the DPSC or BMMC, every day we change the media to keep the cells alive. So what we used to do is we used to throw this culture medium into the trash. We did not realize that that culture medium is having the therapeutic proteins. So first what we did is we collected this, we purified them, centrifuge purified them, and we injected only the culture medium into the hippocampus and studied whether is there any equal neuroprotective effect between the cell transplantation and the conditioned medium injected. So that is what we did. We did a lot of behavior experiment as I explained earlier. As you can see that the red dot is the lesion damaged hippocampus damaged animal, whereas cell transplanted and conditioned medium transplanted, they demonstrated equal cognitive function. There was a drastic improvement or drastic preservation of the cognitive function in this animal, whereas untreated so drastic difference in the learning ability. So this says that the cell transplantation mostly work by producing proteins rather than differentiating into dopaminergic neurons or cholinergic neurons or glutamatergic neurons. They are producing some neurotropic factors. They are producing some, and we can't even come to a conclusion what kind of cocktails they are producing. But they are producing certain tropic factors and other biomolecules which are sufficient enough to regenerate the central nervous system. So a lot of studies are going on using the biochemical method to see what kind of molecules are there, all those things. But our study is mostly of behavioral and that kind of things. So we have a collaborator who are looking into that. So when we sacrifice the animal, we found that in the first panel, you can see this is the normal control where you can see the intact, whereas in the chronic acid group, the CA3 neurons are gone. But when you transplant, you see the intact CA3 regions. Here in the cell transplanted or in the conditioned medium treated, you can see the intact CA3 thing. The very important thing here is there is an enormous increase in the neurogenesis. You can see in the normal control, these few dots, dark dots here, those are the newly born neurons, which can be detected by a protein called double coating. In a kinic acid group, you can see there is a little increase in the neurogenesis compared to the normal control because the neurogenesis is an activity dependent plasticity. You go for an exercise, when you hear a music, when you like trekking, when you go to a hill area, this is an activity dependent thing which can trigger your brain stem cells to proliferate and differentiate. That's why you feel good when you go to this area. So in a kinic acid also, there is an epilepsy, there is an seizure, which is also an activity dependent plasticity. So you can see little more in, but the drastic thing what we observed is that the huge neurogenesis in DPSC as well as in the DPSC condition medium, where you can see the neurons, their branches, the dendritic arborization. This shows that they are super healthy. stem cells derived from human, which are basically from the dental pulp, which are basically from the bone marrow, can actually enrich your neurons, can actually activate your stem cells and make them super healthy. So this we did, and we have recently published this paper too, the Brain Research Bulletin. And now we have shown that the secretome, the conditioned medium itself has the potential. So what is the advantage of this? Here you can use your mesenchymal stem cell as a drug producing factory. You passage them to certain period of time. You just milk this protein, concentrate this protein and administer. It is not a cell transplantation that has to go through a lot of clinical thing, because it will become a product now, a protein product, so that you can directly get the drug approval and you can try it for a clinical translation purpose. So from the cell transplantation, which we injected millions of billions of cells, now we have come to a micro level where the micro level proteins can do these miracles. What is next? Is the whole conditioned medium is necessary? That we have looked into. We centrifuged this conditioned medium. We ultracentrifuged, took the supernatant at various sediment rate, and there is something called vesicles. Every cells produce vesicles. These vesicles carry important message from one cell to other cells. There is one such vesicle called extracellular vesicles, exosomes, which can carry genetic material, which can carry active proteins from the host to the from the donor to the host. So when you transplant a healthy stem cells, it can release these exosomes. The exosome carry all the genetic materials, miRNA, RNA, and other stuff. And it also has some of the essential proteins and active enzymes like SOD and other free radical scavenging enzymes. So you can take these exosomes from this culture medium by processing it. And the advantage of this exosome is that they are nano size. So what is the big deal about nano size? Nano size, you can administer this exosome through the nasal route. The nasal route is the best and easiest way to reach the central nervous system. Because they are having everything, now I can take the exosome from the dental pulp stem cells. I can inject directly to the epileptic subject. No surgery needed, no intravenous, no lung entrapment. It directly goes into the brain region, and it can. This is at the early stage. We have just got 193 lakhs project from DBT to study this clinical effect. We will be, happy to share this data after 1 year or 2 years. But we have done some pilot data where we have collected this exosome, processed them through the ultracentrifuge method, and we have put in a neural culture and see how this exosome get incorporated into the neurons. As you can see here, this is a green fluorescent protein expressed by neural cells. The red color is the exosome labeled by a dye called PKH, and you can see that they goes and bound to the neurons. The green cells, whatever dot you are seeing, actually it is a nucleus staining. And if that is the case, they are in the cytoplasm, they are in the membrane, and they can go and bound to the neurons. So in this model, the in vitro thing, we put the same drug which can induce epilepsy. We put that in the direct cells, and we could able to see the cells are dying. When you treat them with the DPSC exosome or BMMSC exosome, there is a significant neuroprotection. So this is a proof of principle we showed to the funding agency that these nanomolecules, these nanoexosomes can be a potential therapeutic thing for future treatment in neurodegenerative disease. So now currently what we are doing is we are collecting these exosomes. So in a culture condition, you can hear here, you might have heard about 2D culture and 3D culture. The 2D culture is something like it adheres to the plate and it is not as close to our real physiological condition. Whereas 3D culture is something three-dimensional culturing, and the exosome produced by the 3D culture is almost equivalent to the real physiological condition. So right now we are doing an investigation on these 2D and 3D exosomes in an animal model of neurodegeneration to see after the intranasal administration of this in a microliter, how it can travel through the olfactory bulb, get into the hippocampus, where and all they are getting localized, whether they are able to reduce the inflammation, the neurodegeneration, and eventually whether they can reduce the seizure episode in the temporal epilepsy model is what we're studying now. I'll end up my study, my presentation here, and I thank the organizing committee for giving me this opportunity to share my research data. Thank you.
Thank you, sir. Now the forum is open for the questions.
Sir, I have a query here.
Yes, sir.
Sir, is there any chance that studies increased blood pressure because of the
Excuse me, sir. There is some disturbance in the background. Yeah, now.
You can hear me, sir?
Yeah, there is some kind of mic noise is coming from background.
You can hear me now, sir?
Okay, now I can. Yes, now I can hear you.
So what best we can link if there is any mutations related to these epilepsy or with respect to this Alzheimer's and other things, can we link the genes exposed to the fluoride?
Yes. So I don't know about the Indian population, but in the foreign thing, there are certain genes involved in the Alzheimer's disease. They are prone for Alzheimer's disease. Based on that, we have transgenic models. In case of epilepsy, also there is a genetic background to it. But I don't know about the fluoride part, how the fluoride exposure, how the genetic background, how the susceptibility. Probably there should be a study.
So it may cause certain mutations in the genes that cause.
Yes.
So can we do certain studies in cell lines or something like that, and can we
yes,
take it
definitely. So for example, yes. So for example, if you have a neuronal culture and exposing this fluoride, and we can see what mechanism by which it is causing the cell death, what all the gene changes they are taking place. So we can do a complete molecular biology analysis. And then we can take that one and correlate with this human sample to see whether it is matching.
Okay. So I will get back to you, sir. So please.
Sure, definitely. Definitely, sir.
Thank you, sir.
Definitely, sir.
Thank you.
Thank you, sir.
Sir, any questions, sir?
I don't have any question. If any audience want to ask question, I can address it.
Yes, sir.
It's
Thank
okay.
you, sir.
Thank you so much for
elaborate
inviting me.
sir.
Hey. Sure, definitely. I'll sometime we will visit. You can also come to our institute and see our infrastructure.
Yes, sir.
Sure. Thank you so much.
Please visit our center once. Our
Sure, sir.
So if you can please let us know your convenient time, so we can make an arrangement for visit here. Our team also wish to visit your center, sir. I'm an alumnus of
Please come, sir.
2001 to 2004. So
okay.
A lot of affinity towards the market. So sure I will visit.
Sir, please, you are always welcome with your team. I will also visit your research center one time.
Yeah. So I request you to please make a visit to our center and give your reports.
Sure, definitely. Thank you so much, sir. Thank you.
Sir? Am I
Yes.
audible, sir? Yes. Sir,
Yes,
good
yes.
afternoon, one and all. I, Dr. Munilakshmi, take this opportunity to propose vote of thanks on behalf of Department of Biochemistry, Sri Devaraj Medical College. First and foremost, we salute our late founder, President Sri Aral Jalapa, who continues to be a beacon of light. We would like to thank our patrons of Sri Devaraj Education Trust, respected President and Honorable Chancellor Sri G.H. Nagaraja, respected Vice President Sri Rajendra J, respected Secretary Sri Hanmantraju KG, Honorable In Charge Vice Chancellor and Registrar Dr. D.V.L.N. Prasad, Principal Dr. Prabhakar Sir, and respected Medical Superintendent Dr. Krishnappa J. I thank Dr. Shashidhar Sir, for Professor and Head Department of Biochemistry of Sri Devaraj Medical College, for his continuous support and encouragement. I also extend my thanks to other officers of the Academy. I thank distinguished luminary Dr. D. Anandh, Professor and Program Coordinator for Translational Neuroscience, Manipal Academy of Higher Education, Bangalore, for accepting our invitation as speaker and delivering an insightful presentation. Thank you, sir. I thank all the faculty members and stakeholders of SDHE for their enthusiastic participation in this event. Last but not least, I thank the non-teaching staff, photographer and technical team members who are part of this program. Thank you, sir. Participants.
I request all the participants to kindly open their cameras so that we can take on group photo. Thank you, Dr. Harish. Thank you, Anandh.